Welcome back, everybody. Today we're going to be learning about Punnett squares and doing a worksheet in Canvas that is pretty challenging. But it's definitely possible to do it, especially if you let these slides and my voice walk you through the process. This is something you will be tested on in the mastery check, so it's something you kind of need to understand and need to be able to replicate or do. So we've seen these organisms before, and we've talked about how half the genetics from one and half the genetics from the other, when they sexually reproduce through a flower in this case, we get an organism that is a unique combination of both. That particular combination can be predicted, and there is a particular method called Punnett squares that has been used to predict that. So a scientist named Gregor Mendel did studies with pea plants and was looking at purple versus white flowers and trying to predict what would happen, and he came up with this method, and turns out it's super scientifically accurate and helpful, and is still used today. So in our last genetics notes session, we talked about dominance versus recessive, or capital letters versus small letters, right? That's how we show dominance is with a capital letter, and we use a lowercase letter to show recessive. We also talked about genotype versus phenotype, and we learned about 3 types of genotype. So let's review that just a little bit. So with genotype versus phenotype, we know that genotype is the genetic makeup of an organism, or the genes or alleles that the combination of the organism has, right? And this is where we see these alleles represented with letters, and we use a capital letter to show a dominant trait and a lowercase letter to show a recessive trait. We also talked about phenotype, which are the physical characteristics, or what a characteristic looks like the trait that it has that we can see, right? Like curly hair, or tall, or brown eyes, whatever. So with Punnett squares, we're able to sort of use these things to predict outcome. So with phenotype, we can see like this particular offspring, right, from the original slides. This is the phenotype, is this light green color, looks kind of like a cauliflower, but it's green like a broccoli, and so on. So we would say that that's the phenotype. But we want to be able to predict the genotype too, and to be able to look at an organism's parents and predict what the genotype or the alleles are going to be. Punnett squares are the best way to do that, and we know that different types of genotypes include what's called homozygous dominant, meaning that there are 2 copies of the dominant allele, or 2 uppercase letters. Zygous recessive, meaning that there are 2 copies of the recessive allele, or 2 lowercase letters. And then heterozygous, meaning that there are 2 different alleles, and in this case we use R, but like an uppercase and a lowercase. So that would be heterozygous. We can take these alleles from mom and the alleles from dad, and we can combine them and predict what the offspring are going to look like. And we use Punnett squares to do that. So the Punnett square is a diagram that shows the gene combinations from both parents and what the potential results might be in genotype for the offspring. In order to do this, it gives us a probability, or the chances, the percentage chances of inheriting a particular trait. So if we look at this Punnett square here, you can see that it's a square with 4 smaller squares inside. We have 1 organism on the top and 1 on the side. This can be mom, this can be dad, or this can be dad and this can be mom. It doesn't matter where you put them, but like you need to make sure that mom is on one side and dad is on the other. And then we cross them, and we know that we get half from mom and half from dad, and that gives us our new organism. But we look at these different genotypes, and we look at the options, and each square is worth 25%, making the whole square worth 100%. So these are the probabilities of getting these particular genotypes. So if we look at the parents, we have 1 on top and 1 on the side. Like I said, it doesn't matter which is where, but we just have 1 on the top and 1 is on the side, and we have the offspring in the middle. We complete a Punnett square by using those parents and crossing them with each other, one allele at a time. So like here we're going to use large A from, say this is mom, and we're going to bring it down. We're going to use large A from dad, and we're going to bring it over. And in that case, we get 2 copies of large A, or homozygous dominant. Over here we have mom's got a little a, so we bring that one down. But we bring the big A from dad over here, so we get a large A and a small a, and that would be heterozygous. And so on and so forth. You've got this example here with these arrows, this example here with these arrows, and the letters X and Y. This one would represent gender. And we can predict like how many of each genotype we'll get with our offspring with these particular parents if they breed. So let's give another bit of an example. So each parent, in this case, we have big Y, little y with mom and big Y, little y with dad. And we have a chance of giving a big Y or a little y to the next generation in the gamete because it's split up randomly and also evenly. So there's an equal chance for giving either because they have one of each. And so if we cross them, we have like one parent on the top and one parent on the side. Both parents give a big Y here. One parent gives a little y, the other gives a big Y. One parent gives the big Y here. The other parent gives one parent gives a little y here. The other parent gives a little y. So we know with these 3, we're going to get the dominant in all 3. And if dominant is yellow, these are all going to be yellow. We know that this particular one, though, is little y, and that's recessive. Because we have 2 copies of that, we're going to end up with white. So we have a 75% chance of getting a yellow organism and a 25% chance of getting a white one. And we can also see that the genotypes are 1, 2, and 1, meaning that we get 1 homozygous dominant, 1 homozygous recessive, and 2 heterozygous. So now you're going to give an opportunity to do this as a practice. If we have a genotype of big T, and little t, big T being tall, little t being short, we're going to cross these 2 parents together. So we would set up our Punnett square here with big T on one side and little t on the other. And then we're going to go ahead and complete the Punnett square, bringing 1 from each box over in each case. So we will have our offspring predictions from these crosses. And what we see is we see a 100% chance of big T, little t, because we have 25 plus 25, which is 50, plus 25, which is 75, and another 25, which is 100. So we have a 100% chance of getting this genotype for the offspring of big T, little t. Remember that big T is tall and little t is short, and all of these organisms have big T, so we know that all of them are going to be tall as our phenotype, and all of them are going to be big T, little t as our genotype. So you have a couple of other opportunities to practice on these slides. If you want to, you can go through some other examples. There are quite a few here. We talk about some different organisms like horses, straight hair and curly hair. We talk about corn with smooth kernels or wrinkled kernels. We can talk about short hair, long hair in cats. There's a few examples here to practice on. And then you're going to, when you feel like you get it, go to your worksheet and complete the practice there. It is some practice Punnett squares as well as some questions to answer about the different alleles, about dominance and recessiveness, and about probabilities and the chances that you're going to get a particular genotype or the chances that you're going to get a particular phenotype. Remember that each box is worth 25%, and that's how you're going to figure out the probabilities, right? Okay, go tackle it. Do some practice in your slides, and then go, and you don't need to turn that in. It's just a little practice on your own to get the hang of it. And when you feel confident, go ahead and open up that worksheet, make a copy of it, and complete it. Get it submitted into Canvas, and you are a geneticist. Good job. Have a good one.